Hi, my name is Mary Kate Pensevine. Today is June 22nd, 2022, and today I'll be presenting on the regulation of sperm maturation. You can see here in the slide, as we'll move on to the next slide here, we'll be talking a little bit about spermatogenesis first. And what is spermatogenesis? It's a multi-step process of the male germ cell expansion and development. This process occurs in the seminiferous tubules of the testes, and once the sperm is mature, it's transported to the epididymis where it's stored. Testosterone is the main hormone that is essential to the process of spermatogenesis, and we'll get into the breakdown of what testosterone specifically does in this process a little bit later in this presentation. So as you can see here on the right hand side, we have a larger anatomical view of the testes, the epididymis, and then a cluster of seminiferous tubules. The epididymis sort of sits on top of the seminiferous like the vesicle that holds the seminiferous tubules, and that's where the storage of the sperm occurs. In the closer view of the seminiferous tubules, you can see a few things. You see well-formed sperm heads and spermatids in the cytoplasm down here on the right-hand side. And then up here is a Sertoli cell. That is where a lot of the production of the sperm will happen. And then over on the right-hand side, you can see the breakdown of how sperm is produced. You start with the primordial germ cell. Through a few steps, mitosis occurs in its mitotic division. You're left with a primary spermatocyte. The first step of meiotic division occurs next, and then the second step of meiotic division occurs, producing the spermatids, which will eventually mature into sperm. We won't step too much into what that process actually looks like in this presentation, but we'll talk about what regulates it and where it occurs and what the anatomical features of this area of the seminiferous tubules looks like. Moving on, we'll talk about the anatomy of the seminiferous tubules first. There are three major cell types. There's the paratubular myoid cells, also known as PTM, the Sertoli cells, and then the germ cells. The PTM cells they surround the external wall of the tubule and contract to force sperm down the tubule. The Sertoli cells, in contrast, relay external signals and provide factors that are required for the proliferation and the differentiation of the germ cells. They cooperate with the PTM cells to produce the basement membrane, or that surrounding layer of the seminiferous tubules. They provide the niche for spermatogonical stem cells, and that produces the germ cells that will eventually develop into sperm. Down here we have a little image of the myoid cells here on the bottom, again, sort of providing the external wall, the spermatocytes in this layer, the round spermatids, and then the elongated spermatids, just different kinds, and then the Sertoli cell just down here where the development really does occur. We'll next talk about the hormone testosterone. This is the male sex hormone with multiple different regulatory functions. It assists in sex drive, bone mass, fat distribution, muscle mass, and the production of red blood cells and sperm. And the quantity of testosterone in the male body will determine just how extreme some of these factors are in a person. Testosterone is produced in the Leydig cells in the interstitial space of the testes. Testosterone is required for this process of spermatogenesis that we just sort of broke down. And it has a regulatory role, and we'll talk about what exactly it regulates. So the role of testosterone in spermatogenesis is basically broken into four different factors. It functions in the maintenance of the BTB. The testosterone facilitates the reassembly of the BTB components on the basal side of the transiting spermatocyte, so it'll take the broken down components and reassemble them into a new structure in the spermatocyte that's moving. Testosterone also regulates in meiosis. Without testosterone, spermatogenesis would be halted during meiosis and the germ cell would never eventually develop. Similarly, in testosterone helps in the regulatory function of Sertoli spermatid adhesion. Without testosterone, round spermatids are prematurely detached from Sertoli cells, which means that there are no like elongated spermatids, which you can see actually back in this photo here. So we see the presence of elongated spermatids in a lot of the seminiferous tubules, and in the absence of testosterone, those are not able to occur because they're prematurely detaching from Sertoli cells. Testosterone also helps function in sperm release. Without testosterone, the mature sperm cells are retained by the Sertoli and they're not able to be released for their actual function, and they're not able to be stored in the epididymis. So testosterone has a lot of really important regulatory functions, and more so, I would say, the absence of testosterone is felt in these certain ways. These are just a few examples. So we'll talk about the pathways of testosterone. There's a few different pathways. There's the classical and then a few different non-classical pathways. First, with the classical pathway, testosterone will diffuse through the plasma membrane. It'll bind the androgen receptor, causing a conformational change. The androgen receptor is then released from heat shock protein, so it's released for movement. The androgen receptor will then translocate to the nucleus. It'll bind the androgen receptor elements in the gene promoter regions, and then it will recruit proteins to help regulate transcription, and then therefore the production of further proteins. And this is sort of a more straightforward pathway, more easily followed, and the classical pathway when testosterone is able to diffuse through the plasma membrane in that specific site. The non-classical pathway has 2 or 3 different possible processes, but I'll talk about the more streamlined one. In this setting, testosterone would induce the influx of calcium into the Sertoli cells. Testosterone would also cause the activation of G protein coupled receptors and phospholipase C. This will hydrolyze PIP in the plasma membrane, producing IP3 and DAG. This decreased presence of PIP will promote the closing of channels, which cause the membrane resistance and increased depolarization in the cell as a whole. And this depolarization will allow for voltage-dependent calcium channels to open, and calcium will influx, altering the cellular processes. So these are a little bit dense and a little bit complicated, and these are obviously very simplified versions of the pathways that testosterone can follow to make a tangible effect on the cell. But overall, testosterone has a lot of really important roles in the regulation of sperm maturation. It'll follow these pathways. We talked about some of the different areas that testosterone specifically will act on and what it can do, and the really important functions that testosterone or that testosterone plays in regulating the spermatogenesis process. Here are some of my sources. Thank you.